All right, welcome to session number 8. My name is Demba, and I am the chair of this session. It's my pleasure to welcome Dr. R Edmondson, who's going to be our first speaker here. And Laura is going to talk to us about expansion and contraction of resource allocation in sensory bottlenecks. For this session that is going to last until 1 p.m. Eastern Standard Time, we encourage you to ask questions on the Slido. Laura, you can take this away whenever you want.
Okay, thanks. So today I'm going to talk to you about expansion and contraction of resource allocation in sensory bottlenecks. So when we were thinking about this problem, the first thing that we're thinking about is that in many different sensory areas, we have cortical magnification. So you can, if you think about the homunculus from touch, there are certain parts of the body that tend to be more magnified than others. So for example, the hands tend to be over-magnified in somatosensory cortex and the lips of the face. So we wanted to ask kind of what determines this cortical magnification. Is it the density of the receptors, which can change? So these change typically in touch, and they can change in vision as well between the fovea and the periphery. And also the activation of these receptors might be different. So for example, how we use our hands might change. Or is it some combination of both of these that influences the allocations? So we wanted to think about this in a theoretical way because there has been like little treatment about whether it's the density of receptors or the activations of receptors that influences these representations. So in the brain, we have this problem of resource allocation. So if we think about touch, and so here I'm using this example with 2 digits. Over the hand, we have a different density of receptors. So for example, the receptors are much more dense in the fingertips than they are in the rest of the hand. And also the way we use our hands might be different. So for example, here, we might use our index finger much more than our middle digit. So I've shaded this in the dark blue here to indicate this. So how might we then allocate our resources that we have to the 2 different digits? So maybe because our index digit is a lot more active, we might allocate more resources here shown in the dark blue to this digit, whereas with the middle digit it's activated less, so maybe we offer less resource allocation. In terms of the density, because these receptors are, because there's many more receptors there, we might offer more allocation to these as shown in the orange. So one thing to think about when thinking about the resource allocations is that when the receptors are more dense, they correlate, their responses correlate a lot more with each other. So the covariances are higher if you're with your neighbor than they are with receptors that are more distant from you. And we can also model the change in the activation by just scaling this covariance function. So in the brain, we have this problem of sensory bottlenecks, and we can have an explicit bottleneck or implicit bottleneck. So an implicit bottleneck is where there are a lot more input neurons than there are output neurons. So for example, in the optic nerve, we have many input neurons, but the optic nerve acts as a bottleneck because there are fewer outputs, so things have to be compressed there. For an implicit bottleneck, the output neurons simply don't represent all the information in the input neuron. So this applies some kind of limit on the amount of information that we can transfer. So we think about this problem through an efficient coding framework. And these kind of efficient coding problems ask questions like, how should a group of neurons encode the information in our inputs? So here they suggest that we should maximize our information transmission. And one way to do this is by reducing the redundancy between neurons. So we're going to restrict ourselves to second order statistics. So it's going to be a decorrelation model. And these models have been very successful in explaining certain things about sensory processing, so like in visual processing and auditory processing. And in our model, we're going to decorrelate using principal component analysis. So the way we model our information bottleneck using this is by simply retaining fewer components. So we retain the components with the highest eigenvalues, and this allows us to set a limit on the amount of total information. So if we have a very small bottleneck, we're going to retain fewer components. And when we have a large bottleneck, we're going to retain more of the components. So normally in these efficient coding models, you ask questions about what the receptive fields look like. But in our case, we're instead going to ask, how should the receptive fields be placed? So if we have many regions, how should we allocate our receptive fields? Which region should we put them in? So in order to do this, we actually don't need to think about the eigenvectors. We only need to consider the eigenvalues. So now I'm just going to explain how the model is set up and how we calculate these allocations. So imagine that we have two regions, and our first region is 3 times as dense, here shown in the blue, than the orange. So we can then calculate the covariance matrices for these two regions. So this is based on their responses. So in our formulation, we use a negative exponential covariance matrix because this allows us to solve the problem analytically. So we can then calculate the eigenvalues of those two matrices, and then we're simply going to sort the eigenvalues from the two regions. So as you can see here, we have in orange the eigenvalues from the low density region, and in blue the eigenvalues from the high density region. So in order to calculate our bottleneck, we can simply take a subset of these eigenvalues, so they're ordered by the magnitude as well. So if our bottleneck is, say, 30% of the total possible information, then we can simply count how many of these eigenvalues adds up to 30%. So here we can retain 10 eigenvalues, and then we simply calculate how many eigenvalues we have that are from each region. So we can just sort them and count them. And in this case, we can see that the allocation from the two regions at about a 30% bottleneck should be 30% of our resources should be allocated to the low density region, and 70% should be allocated to the high density region. So this is the first result, and our result is that the resource allocation really depends on the bottleneck width. So you can see in this plot that as we increase the bottleneck width, the allocations change. So here we have the allocations from the low density region in orange and the high in blue as before. So first you can see that the allocation actually changes in a non-linear way depending on what our bottleneck width is. So if we just take a closer look at different stages of this bottleneck, so imagine that we have a very narrow bottleneck, for example, around 5%, compared to a proportional allocation, so just allocating according to the densities in each region. You can see that we actually have no representation of the low density space when this bottleneck is very narrow, and it is completely dominated by the high density region. So we term this as a contraction of the representation of the low density region. For an intermediate bottleneck, we actually see an increase in the amount of resources that we allocate to our low density region. So here we have what we call an expansion of the resource allocation. And this converges to a certain limit that we can calculate from our analytical solution. And finally, for a very wide bottleneck, so here around 90%, you can see that the allocations are returning back down to a proportional allocation again. So how are these allocations influenced by both the densities and the activations? Well, we can think of looking at these two things separately and then together. So first, I'm going to go through what happens if we hold these activations between the two regions constant, but we change the densities. So here we have our two regions again, and we have a low density region that's shown in the orange and a high density region that's shown in the blue. So if we change the densities, we're going to see an expansion of the low density region and contraction of low density region depending on the width of our bottleneck. So both of these scenarios are possible. If we hold the density between the two regions constant, but then increase the activation, say on this orange region, then we'll see a sole expansion of that region for the whole bottleneck. Or if we increase the activation on the other region, we'll see that this region actually contracts its expansion for the entire bottleneck. So in terms of the densities and the receptor activations together, if these tend to be positively correlated with each other, so we have high density and a higher activation in that region, then this orange region here is either going to be contracted for the whole bottleneck or expanded and contracted. So we see sole contraction when the positive correlations is quite strong, and when it is weaker, we see both an expansion and a contraction. So if the density and the receptors activations are negatively correlated, so we have a low density region with a lot of activation, then our orange low density region is going to either be expanded for the whole bottleneck if this thing is quite a strong negative correlation, or if it's a weaker negative correlation, we see expansion and contraction. So overall, our resource allocation really depends on our bottleneck width. So these input regions can expand or contract compared to their densities, but this exact profile of expansion and contraction is affected by both the densities and the activations. So then we decided to think about what might our model suggest about plasticity. So imagine that we have a constant receptor density, so these don't seem to change over time. And our bottleneck width is also constant, so the amount of resources that we have doesn't change. But our stimulus statistics can maybe change over time. So this might lead to a reallocation, or you could see this as plasticity. So for example, if we have our two digits again, our middle and index digit, at a very narrow bottleneck, maybe the index digit has a much larger representation than the middle digit, and at a slightly wider bottleneck, the middle digit has a slightly larger representation. If we now stimulate the middle digit a lot more, then we might see that the allocations actually increase for a narrow bottleneck, so that they might increase quite a bit. But in a wide bottleneck, the allocations for the middle digit maybe don't increase so much. So more generally, this can be seen in this plot here. Whereas if we increase the amount of receptor, the amount we activate the region, so here we increase quite a lot, we see a higher or a greater difference in the allocations for very narrow bottlenecks shown in the green than we do for wider bottlenecks, say in the purple. So overall,
Laura, Demba here. Just a heads up that you have a couple of minutes.
That's fine. So in a very narrow bottleneck, we tend to see higher plasticity, so that can be seen here. And in a very wide bottleneck, we tend to see lower plasticity. So then we decided to think about how we could actually test this empirically. And so we found this model system that's called the star-nose mole. And this model system has been extensively documented by the Catania lab at Vanderbilt. And so this creature called the star-nose mole, it tends to live underground, so it relies a lot on its sense of touch rather than vision. And in order to feel for its prey when it's underground, it has this has two sets of 11 like tactile rays, they're called. And the densities of the receptors in these rays varies depending on which ray we're looking at. And also how it uses the rays changes. So as it's feeling for the food, it tends to funnel the food from the outer rays towards ray 11, which is next to the mouth. So the Catania lab has also extensively documented the cortical allocations here, and we can see that actually this ray 11 has a much larger allocation in cortex than the other rays do. So then we wanted to see if our model could predict these allocations well. So we decided to test 3 models, and we tested our model with including only the accurate densities. So that can be seen here. Then we decided to test another model where we included only the accurate receptor activations, but we were going to keep the density the same. And that can be seen here. So here we have a slightly better prediction. But when we included both the activations and the densities, our model gives the best prediction of the cortical allocations, which can be seen here.
Laura, if you could wrap up, please, I'd appreciate it. You have quite a few questions here.
So in conclusion, both the receptor densities and the activations determine how the brain should allocate neurons to represent the different input regions. The allocation is non-linear, and this depends on the total amount of resources that are available. The same region can contract or expand depending on different resource constraints. And the strength of the sensory plasticity can also depend on the resource limit. So if we have a narrow bottleneck, we might see higher plasticity than a wide bottleneck. And finally, decorrelation has been shown as a strong driver in sensory processing. But yeah, future work, we would need to consider other things such as noise, or we can consider non-linearities as well. So if you're interested, we had this paper in Neurips in 2019 that you can check out, and we're also soon going to have a preprint out. Finally, I just want to thank my co-author Alejandro and my supervisor Hannah Saal. Thank you. Any questions?
Thank you, Laura. Thank you. We've got a few questions here. Let me go with the first one here. Jan Drogovic is asking, could you provide some intuition for the nonlinear relationship between allocation and bottleneck width? Why is it not simply monotonic? Does that make sense? Yeah. Mm-hmm.
So we actually thought that the allocations would just be proportional depending on the densities. So we actually found it quite surprising ourselves that we see this nonlinear allocation. So I think particularly at narrow bottlenecks, I think we see this like the underrepresentation or like no allocation of the low density region. I think it's to do with the drop off in the covariance function. So maybe if the receptors are really not very correlated with each other, there's going to be a lot more information, I think, in the high density region that comes through earlier on. And then in terms of the convergence towards the limit, so we see that actually in all the different bottlenecks, like intermediate bottlenecks that we have over like very different density and activation combinations. Awesome. And then for the very wide bottlenecks, so we see this kind of drop off at the end where the high density information, the information from the low density region has actually run out. And so there's only information left in the high density region to allocate.
Awesome. Awesome. Thanks. We have time for one more question, and I'm going to ask to bring the next speaker. How do your results change if you modify your definition of resources to say information or entropy or the number of spikes or whatnot? So how much does this depend on you having picked variance basically in terms of these eigenvalues as opposed to something like information or entropy?
Yeah, so I think, so in terms of ours, we define it as this like explicit bottleneck. So we have the number of resources that we have is fixed. If we think about it in terms of, say, the total variances instead, I think we would see a similar shape with the non-linearities, but where this is in the bottleneck might change. So we don't actually consider spiking in this, but it's something that, I don't know, maybe could be considered. But in terms of our setup, yeah, we don't consider the spiking in terms of the spiking information.
Yeah. Thank you, Laura. We have a few more questions. Unfortunately, I don't have time to ask them. I would encourage people whose questions were not answered to connect with Laura offline. Thank you, Laura. I'm going to invite the next speaker. Thank you.
Thank you.
Hi.
Hello, James.
Hey, how's it going?
It's going great. It's going great. Thank you. Thank you for being here. And I'm looking forward to hearing your talk. and you're going to tell us something about the dimensionality of neural activity in sensory cortex.
Can you see my screen?
Yes, I can see your screen, and you can take it away whenever you're ready.
Great. So thanks a lot for the introduction, Demba, and hi, everyone. So I'm James Rowland. I'm a PhD student at the University of Oxford in the group of Adam Packer, and today I'm going to be presenting our experimental work on the dimensionality of neural populations in sensory cortex and its relationship to both propagation of activity and to behavior. So the key question I'm going to try and address today is, how does the dimensionality of a neural population impact propagation of activity between distinct cortical regions of the mammalian brain, and does this have an impact on behavior? So why is activity propagation important to study? So we've actually known that reliable propagation of neural activity between brain regions is critical for healthy brain function for well over 100 years. And this goes back to the pioneering work of Wernicke, who showed that every brain region along this pathway, so the medulla, the auditory cortex, and the insular cortex, were critical for the production of speech. And disruption to any brain region along this pathway, or the communication between them, prevented regular speech. So why is dimensionality why is dimensionality of a neural population an important feature to study? Well, it's been shown many times that projecting neural activity onto a low-dimensional space can reveal how it encodes information. And this is demonstrated here by this beautiful example from Gilles Laurent's lab, who showed that these low-dimensional projections explain how neurons in the antennal lobe of the insect olfactory system encode both the identity and the intensity of an odor. And it's also been shown that the effective dimensionality of a neural population is likely critical to its ability to represent information. And this was shown particularly well recently by a tour de force from Stringer et al., who analyzed the eigenspectrum of population activity. And through simulations, they show that in a highly correlated population, a single eigenvector can explain almost the entire variance of the activity, and the population exhibits low-dimensional dynamics. Whereas in the other extreme, a population of independent neurons, all eigenvectors contribute equally to the variance, and the population exhibits high-dimensional dynamics. But in actuality, the brain exhibits dynamics somewhere in between these two extremes, thereby preventing small changes in input from dominating the activity of a population. And effective dimensionality has also been linked to attention, arousal, and behavioral performance, thus motivating the study of its relationship to activity propagation. However, the study of activity propagation is challenging due both to technological constraints and due to biology. However, the recent renaissance in optical methods enables us to study propagation of activity in vivo in a behaving animal with single-cell resolution. However, there are a number of issues we have to solve to enable this. So the first issue is recording simultaneously from two brain regions in a mammal. And we can solve this by employing a 2-photon microscope with an optical path that facilitates imaging a large field of view while maintaining decent spatial and temporal resolution. Second, we need to ensure that the activity we're recording is actually being propagated between our two brain regions, and they're not just correlated due to sharing a common input. And we can do this by taking experimental control of neural activity and driving spikes artificially through photostimulation. So if we photostimulate one brain region while recording from another connected region, and we see activity in the second brain region locked temporally to our photostimulation, it's very likely that we're seeing propagation of activity rather than common afferent input. And finally, we want the propagation of activity to not just be passive, but to be behaviorally meaningful to the animal. And we can do this by training mice through operant conditioning to associate the photostimulation with reward. And I'll get onto how we do that in a few slides' time. So how do we make a prep that enables us to optically read and write neural activity across brain regions? So we do this with a dual viral injection strategy where we inject GCAMP6S, a calcium indicator, and a red-shifted opsin, C1V1, which allows us to perform photostimulation without too much optical crosstalk with our imaging. And we target these viruses to the border of S1, the primary somatosensory cortex, and S2, the secondary somatosensory cortex, which sits upstream of S1 with respect to the flow of activity from the whiskers, while being densely reciprocally connected with S1. And we then install a glass window to enable optical access across S1 and S2, with the aim to see expression of GCAMP that spans across both brain regions. And we localize precisely the two regions by stimulating individual whiskers during wide-field calcium imaging, shown here, with the inset, the white box, showing the field of view that we use to perform single-cell imaging. So our goal when we do these experiments is to photostimulate neurons in S1, and only in S1, but while recording the activity both of S1 neurons and S2 neurons, allowing us to get a handle on how activity propagates between these two sensory regions. So how do we actually perform our photostimulation? So to do this, we employ two-photon photostimulation, which enables us to photostimulate groups of individual visually identified neurons without activating neighboring cells. So this is shown here in this cartoon depiction of the experiment, where the goal is to target these three neurons here, and only these three, but while recording the activity of the entire population. So the reason why we employ this method is that it allows us to inject more physiologically relevant patterns of activity than conventional wide-field optogenetics, which would cause every neuron in the field of view expressing opsin to become activated simultaneously. And the other advantage of this technique is that it allows us to parametrically vary the number of cells we are stimulating on a trial-by-trial basis. And the reason for varying the number of cells stimulated trial by trial will become apparent in a moment. So what does this actually look like in practice? Well, here we have a highly processed example movie where we're jumping our photostimulation beams around the field of view and sequentially stimulating small groups of neurons. So here we're stimulating neurons that are spatially co-localized, but in practice, we can stimulate groups of neurons that are spread up to 350 microns apart. Okay. How do we ensure that our propagation of activity is not just passive, but rather has behavioral meaning to the animal? We do this by training mice through operant conditioning to learn the association between optical activation of S1 and reward. So after the initial training, we photostimulate groups of neurons in S1 and ask the mouse to tell us if it perceived the stimulation. And if it perceives the stimulation, the mouse is trained to lick a spout for a water reward. So this intentionally non-naturalistic task has an advantage over classical sensory-guided behavior tasks because we know that the activity we're recording in S1, as well as the regions S1 is connected to downstream, are causally driving the behavior. So we have two different trial types, go and catch trials. On go trials, we photostimulate groups of neurons in S1. If the animal responds to the stimulation by licking, we score that as a hit, and the animal is rewarded. If the animal fails to perceive our stimulation, we score that as a miss, and the animal receives no reward. And then we have a second trial type, the catch trial type, in which we don't perform any stimulation, but the structure of the trial is exactly the same as go trials. This is to ensure that the animal is not just getting rewards by licking randomly, but is actually perceiving the stimulation. So our goal when we design the experiment is to perform photostimulations that span through the perceptual threshold of the animal. So on some trials, the stimulation is perceived, and on some trial it's not. And we do this by varying the number of cells stimulated on a trial-by-trial basis. And this allows us to generate a psychometric curve of the animal's performance against the number of cells we photostimulate. And animal performance is quantified using the D' metric, which tells us how much more likely the animal was to lick on go trials versus catch trials, where a D' of 0 is chance. And as we can see, the animal is able to reliably detect activation of between 30 and 40 neurons. So we're not only photostimulating neurons in S1, but we're also imaging the resultant activity, both in S1 and in S2. And from this, we can plot the average population response to each trial type. So this is a time series averaged across cells, across trials, and across sessions. And the gray box here indicates the time during which we're photostimulating. And this is blanked because the laser causes a large artifact. So on hit trials, when the animal perceives the stimulation, we see a greater activation of S1 than on miss trials, which is unsurprising given that we're directly driving spikes in this region. However, strikingly, we also see robust activation of S2, so we're getting reliable propagation of our perceived stimulation from cells that were directly targeted with light in S1 to the connected region S2. And this is followed by a prolonged period of inhibition. So next we wanted to know, why are some of our stimuli perceived and elicit a hit, and why are some not? Why are some of our stimuli propagated reliably out of S1 to drive licking behavior, whereas some stimuli are either controlled by inhibition or else not behaviorally salient in some way? Well, one thing that might be controlling this is the ongoing activity in S1 immediately prior to the stimulation arriving. So there may be some kind of network activity existing before the stimulus arrives that helps the upcoming stimuli to propagate, and some pre-existing activity that prevents the upcoming stimulus from propagating. And we get an early hint that this may be what's going on through this simple logistic regression classifier. So we show the classifier neural activity in S1 before the stimulus arrives and ask it to tell us whether the upcoming stimulus is going to be a hit or a miss, without it ever seeing any information about the upcoming stimulus or the neural response to it. And we find that this classifier is able to perform above chance, showing that there is something in the neural activity in S1 prior to the stimulus that assists or blocks the upcoming stimulation. But what is the neural activity that exists in the network that may underpin this? What features of neural activity predict whether the upcoming trial is going to be a hit or a miss? Well, there are a number of neural features that one could focus on. Two of the most obvious features are the mean activity in the population. So if we average the amount of activity across all neurons, is this indicative of an upcoming trial? And the second is correlation. So what's the average pairwise correlation between neurons in S1? And we find no evidence of trial prediction based on these two quite straightforward neural features. So we need to look a bit deeper. So as I alluded to previously, the one feature that I'm going to focus on today is effective dimensionality. And this is motivated as effective dimensionality has previously been linked to attention, arousal, and behavioral performance.
James, just a heads up that you have about a couple of minutes left for the talk component.
So as shown by Stringer et al., the effective dimensionality of a system can be computed through its eigenspectrum, where a low-dimensional population has a very high proportion of its variance explained by the first principal component, whereas a very high-dimensional population has very little explained by its first principal component. And we use this to measure effective dimensionality in our data. So we're interested in the single trial level because we're trying to understand what causes an upcoming hit or miss trial from the activity in S1 prior to stimulation. And we believe that the effective dimensionality of a population is dynamic and can change on a trial-by-trial basis. So we take our individual trial and run PCA on the neural activity matrix of S1 prior to stimulation. And from this, we can generate an eigenspectrum on which our data lies, as expected, somewhere in the middle of the high and low dimensional population in these simulations. And we then approximate the effective dimensionality of our population by taking the variance explained by the first PC. So the higher this is, the more low dimensional the population. And if we do this on every trial we've recorded, we can then assess the effective dimensionality in S1 before a hit and before a miss trial to assess whether dimensionality prior to the stimulation has an impact on the ability of the mouse to detect an incoming stimulus. And we find that our propagation is significantly more high dimensional before a hit than before a miss, implying that dimensionality may play a role in assisting the upcoming stimulation in becoming behaviorally salient. So we're not only interested in whether effective dimensionality predicts behavioral outcome, so it may be indicative of attentional state, for example, but we also want to know whether it assists propagation of activity out of S1 and over to connected brain regions. And we indeed get a hint that this is the case. So the higher the dimensionality of S1 prior to stimulation, the more activity gets elicited in S2, implying that dimensionality may be an important feature to study, as it seems to be involved both in behavioral salience and in activity propagation. So to sum up, we've developed a prep to study behaviorally relevant propagation of activity between cortical regions. Reported stimulation of S1 elicits robust activation of S2. And the dimensionality of S1 prior to stimulation predicts both behavioral outcome and propagation of activity to S2. So some acknowledgements. So a big shout out to Rob Lees, who was instrumental in the development of the S1, S2 prep. And then to Thijs van der Plas, Matthias Loidolt, Jonas Dehning, and Viola Priesemann, who've been instrumental in the data analysis side of things. And then to Adam Packer, who's been instrumental in every aspect of the project, as well as its design.
Thank you, James.
Thanks very much. And finally, Packer Lab is hiring. So if you're interested in these kind of questions or in the claustrum, then please get in touch. Thanks a lot.
All right, I think we've got time for a couple of questions. I'm just going to get into them. The first one asks, James, did you monitor arousal, such as pupil size or other behavioral factors, such as whisking, in the pre-stimulus period? Because these might predict pre-stimulus activity and hit or miss.
Yeah, so we haven't done pupil tracking just yet. We're actually in the process of setting it up just now. Yeah, so I think it's been shown in previous work that arousal causes more high-dimensional population activity. So we would definitely expect that arousal, pupil size, whatever it is, predictive of the upcoming hit or miss trial. But yeah, we don't currently have that data to speak to that. But yeah, it's a great question.
All right, thank you. The following question asked, does the psychometric curve depend on whether you activate co-localized neurons or neurons that are spread out in space?
Yeah, again, this is not something we've explicitly tested. It's something we've thought about. So the actual identity of the neurons that we stimulate is randomized on a trial-by-trial basis. So we have every neuron in the field of view, and then on an individual trial, if we're stimulating, say, 5 neurons, then 5 neurons are selected randomly from the groups that can be stimulated simultaneously. So yeah, so we do have a variety of different kind of spatial co-localizations in different trials, but we haven't explicitly looked at it yet, and we didn't explicitly design the experiment to test it. But yeah, it's a great question.
Thanks for the clarification. Next question asks, can animals be trained to discriminate 2 different patterns of stimulation as opposed to detection of one?
Yeah. Yes. So this has been done with wide field optogenetics, so just stimulating 2 groups of neurons with wide field optogenetics, and I also believe it's been done in monkeys. So I think the answer is yes, definitely. I imagine it would be a lot more difficult to train than on this task. But yeah, I think if you had a specific question that required animals to discriminate 2 different kinds of activation, you could definitely do that. Yeah, but we haven't looked at it explicitly.
All right, great. Thanks for clarifying. And I think we've got time for a couple of more questions. Do you see any whisker movement that is induced by your S1 stimulation?
Yeah. Again, this is something that we haven't looked at. So, but we have thought about. So I think like the kind of broad question with this is, what is the animal actually feeling? So we can't really answer that. So from the mouse's perspective, does it feel like, we've activated S1? Does it feel like there's something in its whiskers? And will it whisk in response to that? I would imagine that the answer would be yes. Like I would imagine that if we stimulate whisker S1, the animal would feel like there's something in its whiskers, and they do tend to whisk in response to that. But again, this is something we're setting up alongside the pupil tracking, and we want to check. But I do believe they would whisk in response to the stimulation. Yeah.
Thank you for your talk, James. I'm going to encourage everybody whose questions I couldn't get to connect with James offline. Thanks a lot, James. I'm going to welcome the next speaker. Thank you. Hey, Andrew.
Hi, how are you?
Doing very good, Thank you. How about yourself?
Good, doing well. Can you see my slides
Excellent.
here? Yeah, okay.
Yes, I can see your slides. It's my pleasure here to welcome Andrew Francl from MIT, who's going to tell us about some of the work that they've done on biological sound localization. Take it away, Andrew.
All right, great. Thank you, Demba. So as Demba mentioned, my name is Andrew Francl, and I'm a graduate student in Josh McDermott's lab in the Department of Brain and Cognitive Science at MIT. And today I'm going to talk to you about how optimization for real-world environments helps explain biological sound localization. So many aspects of perception are believed to be adapted to the natural world. For example, in much the same way a scientist may suggest that giraffes evolved longer necks because it's helpful for reaching leaves in higher trees, similarly, pit vipers may have evolved the ability to perceive infrared radiation because it is beneficial when hunting small, warm-blooded mammals. So both of these sorts of theories are intuitively satisfying and fit well with the evidence we have immediately on hand. But these sorts of theories are often incredibly difficult to test. And that's because in order to gather the sort of causal evidence we often rely on when evaluating a hypothesis, it would require us to simulate an alternative environment during evolutionary development. Additionally, many aspects of perception and behavior rely on other aspects of the organism as well, rather than just evolutionary pressure from the external environment alone. So some examples of these are neural implementational constraints and an organism's longer-term goals. It's also worth pointing out here that analogous questions come up all the time in human perception, but are difficult to test for similar reasons. So in order to make some progress on this problem, in this work we propose using supervised machine learning to reveal characteristics of systems adapted to environmental constraints. And this approach is somewhat similar to ideal observer theory, where in ideal observer theory, researchers analytically derive optimal solutions under simple sets of constraints. But unfortunately, this approach is often not possible in more complex problems. So our approach instead relies on optimization and applies to a larger set of complex real-world problems, but isn't provably optimal. And nonetheless, we'll show that this approach can still be pretty fruitful for the sorts of questions we're trying to ask. So we operationalize this approach using a sound localization task. Specifically, this proposal has two pieces. The first is the environmental simulator, which takes a set of latent variables, such as the environment geometry, environmental physics, and the spectrum of a source, and then uses these to render an auditory scene. And then the second component of our approach is a machine learning system that takes that auditory scene as input and learns to use it to predict the source location in that specific scene. So for those of you unfamiliar with biological sound localization, it's an interesting problem for a couple of reasons. First, unlike in vision, the sound location isn't explicitly represented in the sensory input. And additionally, waveforms are all summed when they reach the ear, and that's shown here on this left figure. So that the information you might use to determine the location of a sound is corrupted by the competing sound sources and reverberation. And that the reverberation are all these echoes. So overall, this is in the real world, this is a really complicated problem and presents a complex computational challenge. However, significant progress has been made on understanding how humans do this over the past century, and specifically, three main cues have been put forth is likely to underlie this ability in humans. The first two are the inaural time and level difference, and these are jointly known as the binaural cues, and they're calculated by measuring the difference in arrival time and volume between the left and right ear for a given signal. And the way to think about this is to imagine a single sound wave emanating from the speaker and moving through space, and it's going to make contact with this right ear, but then it's going to have to travel some distance to make contact with the left ear. And that's, of course, going to cause a time delay, and that's detectable by the auditory system. In addition, the sound wave is going to have to move around the head and kind of through it to reach this left ear, and that sort of causes what's equivalent to an acoustic shadow, and that causes the ILD, and that's also detectable by the auditory system. So there's this third type of cue known as the monaural cue, and it's due to the pinna, which is this floppy piece on the outside of your head. And what it does is introduce different notches in the frequency spectrum of the signal based on the position of the source. However, determining the location of these notches is, of course, another ill-posed problem because the initial spectrum of the sound source is also unknown. So at this point, it's worth pointing out that a model that tries to directly calculate these cues to determine the location of a sound works pretty well in simple scenes, but performs really poorly in complicated scenes when things like background noise and reverberation are present. So basically real-world scenes. So after that brief orientation, we now pose the driving question for the rest of this talk. And that's, can we build a model that reveals characteristics of systems adapted to their environment? And we'll break this into three main points. So first, can we build a model that is both fit to our environmental simulator and operates effectively in a natural environment? Second, just because a model works doesn't necessarily mean we have properly constrained the model to work the same way as humans. So next we ask, does this model replicate aspects of human perception? And then third, if it does replicate aspects of human perception, can we determine which aspects of the environment really drive these results? So we'll start with question 1 and a brief outline of how we created this model. So we started by building sets of natural sounds and background sounds. And then to create a single auditory scene, we randomly sample a natural sound as well as a variable number of background sounds. We then render these into an auditory scene using a virtual acoustic world where we control many aspects of the environment, including the room geometry, the room materials, the positions of the sources and background sounds, position of the listener, and many other aspects of environmental physics. And the simulator then produces the audio for the left and right ear that a listener would hear in that configuration with those specific sounds. This audio is then fed through a simple cochlear model to the output, and then the output is fed directly to a neural network. And the neural network has one job, and that's to predict the sound source location of that natural sound in the given auditory scene. So we train the model, and it learns to localize natural sounds from its validation set. But that, of course, leaves open the question of whether or not it will generalize to the real world, and whether or not our virtual acoustic world has adequately captured the important aspects of a realistic environment. So to test this, we made a series of recordings in our lab space using a Kimar mannequin, and that's pictured here in this right panel. And we recorded 100 sounds at 12 different locations. So 50 of these were speech clips, and 50 of these were natural sounds that were not included in the training or validation set. We then took this binaural audio and ran it through our model. And as you can see, our network generalizes quite well to the real world. And our network performance is shown here in the middle. This is a violin plot or a distributional plot. And human performance is shown on the left just for reference. Actual position of the sound sources is shown on this x-axis, and judge position on the y-axis, where perfect performance is across this diagonal here. So now that we see this, we seem to have a model that can be fit to an environment we control and generalize as well to the real world. However, its accurate performance on real world stimuli doesn't necessarily mean it's using a similar localization strategy as humans. So next we ask if this model is adequately constrained to mere aspects of human perception. And to test this, we're going to run psychophysics experiments or behavioral experiments from the prior literature on our trained model. Specifically, we reproduce the stimuli from the original papers and then calculate the network judgment for each of these stimulus. We then aggregate the data to create the same graph previously calculated for humans. It's also worth noting that at this point, the network weights are frozen, so no learning or fitting to the human data at all is occurring. So here we show a set of such experiments where human results are on the top row and network results are on the bottom row. And we're not going to have time to go through all these in detail, and I'll instead give you a quick summary of each. But we hope your overall takeaway message from this is that a wide range of human behaviors are closely mirrored by a model optimized to localize in a natural environment. So going from left to right, we first have evidence that both the network and humans judge sounds more accurately along the midline, and performance decreases as sources move laterally. Next, we show that both models and humans integrate spatial information across frequencies in a similar manner. Third, our model replicates the frequency-dependent use of interval time and level difference cues that is seen in humans. This is also known as duplex theory and is considered one of the most fundamental results in the binaural literature. Fourth, humans and networks show similar sensitivity to the spectral resolution of spatial cues. Fifth, both humans and the networks show elevation localization collapses, but azimuth localization is maintained when the ears are manipulated, as shown in this lower panel here. And lastly, we have similar performance on the precedence effect, which I'll describe in more detail in a later part of this talk. So again, what we hope you take away from here is that a wide range of human behaviors are closely mirrored by optimizing performance in a natural environment. And so now that we know that our model actually does mirror a wide range of aspects of human perception, despite never being directly trained to do so or fit to human data, this now brings us to the last section of our talk. And in this, we'll investigate how the environment drives these perceptual results. So to look into this, what we do is modify some aspect of the training environment and retrain this model in this alternative environment. So these modifications include things like removing echoes from the environment, shown on the top, or removing background noise from the environment, which is shown here in the middle, or training with unnatural sounds, and that's shown on the bottom. And in this case, the unnatural sounds are bandpass noise. So For each training condition, we then measure a normalized average error between the network and human psychophysical results. And then we're going to plot this on the graph to the right. And when we do, what you can see is that when we deviate from natural conditions during training, the model deviates more from human data. So this graph provides a fairly high-level view of the results, but we can drill down much further. And in doing so, we can link specific aspects of perception to the environment using this approach. So in this talk, we'll focus specifically on how we can do this with the precedence effect, which I'll briefly explain now. So in a classic version of the precedence effect psychophysics experiment, a human subject is placed in an anechoic room with two speakers located at positive and negative 45 degrees to their left and right. And each speaker then emits a single click with a variable time delay between them. So if the clicks are emitted simultaneously, then the subject perceives a single click originating directly from in front of them. However, at short time delays, the subjects only perceive the leading click and perceive it as originating at the position of the leading click speaker. And then as time delays increase, subjects are then able to hear both clicks separately, and then at even longer time delays, they're able to accurately identify the location of each click. So as previously seen, our network also exhibits this strong preference for the leading click at short time delays, and then that preference degrades over time. And a natural question is, of course, why do humans display this behavior? And one theory is that precedence effect is an adaptation for localizing in environments with reflections. And the idea here is that if you're trying to figure out where a sound is coming from, you might rely on the fact that the shortest distance between a source and receiver is the direct path, right? And so what you might do is rely on the fact that that sound is going to arrive first and all these echoes are going to arrive slightly later. And so you could pay attention to the sound that arrives immediately and calculate your location based on that, and then ignore all the later sounds that arrive because they're probably echoes. So this is one of those theories that's intuitive and fits fairly well with the immediate evidence we have on hand, but is incredibly difficult to test in some sort of causal way because it would require us to simulate some sort of adaptation that we can't do. In addition, there are a series of alternative hypotheses, including things like resolving the integral phase ambiguities from the ITD. So it's not to say this is, an open and shut case. As mentioned previously, we were able to retrain our model in an environment without echoes, so we can use that model here to directly test this hypothesis. And what we're going to do is use that model retrained in an anechoic environment and then retest using the exact same stimuli that we had just previously shown you. And what you see when we do that is that the model diverges from human results when trained in the anechoic condition. And just to orient you to this plot, this purple line here is the anechoic condition. The blue line is the normal condition. And there are two alternative training conditions that I had previously described, shown in pink and orange. And what's interesting is that it's only in the case where there are no echoes in the environment that the precedence effect really breaks down. Otherwise, it's by and large replicated. So this both suggests evidence that the precedence effect is in fact an adaptation for localizing an environment with reverberation. And it also suggests a new method for demonstrating adaptiveness of perception to specific constraints. So we can...
Hey, Andrew, just a heads up that you have a couple of more minutes.
Yeah, that's perfect. Thank you. So we can generalize this approach and look at each experiment in each training condition, and that's what's shown here on this plot on the right. So on the x-axis, we have various training conditions, while individual experiments are represented by the colored bars. And then on the y-axis, we have a measure of fit to human data relative to the normal training condition, where positive values mean the new model is less similar to humans than the normal model. And I know there's a lot to take in with this specific figure, but what I'd like to draw your attention to is that for each training condition, different experiments seem to deviate from human results. And that's shown by different bars really peaking here. So you see here the precedence effect, bandwidth stimuli, and changing years. And so in each case, something different is driving these results. And this suggests that different aspects of the environment are constraining different parts of perception, and that they seem to be able to be identified. So in conclusion, we successfully trained neural networks to localize sound sources in a real environment. And then we showed that that model replicates a range of human psychoacoustic effects. And then lastly, we showed that training conditions had dramatic and interpretable effects on these localization strategies. If you're interested in more details or want to know more about any aspect of these experiments, all this work is presented in much more detail in our bioRxiv paper. And in addition, our model is available on GitHub. So lastly, I just want to thank Josh McDermott, my advisor, and the entire McDermott lab for their help and support throughout this project. In addition, I want to thank the NSF and NIH for their funding. And lastly, I want to thank you for your time and attention. So thank you very much, and I'm happy to take any questions you might have.
Hey, thank you, Andrew. You give us an extra minute here for questions. So the first question is from Tatiana Engel, and she asks, do humans use active strategies to localize sounds, such as people turning their heads? And if they do, how does this affect
really
comparison with the neural net?
It's not totally clear how active the strategies are and are not. There are a few places where we're pretty sure they do use active strategies, and one is in resolving front-back confusions. So what you do is by turning your head, you basically get the derivative of the ITD and ILD. And so that's definitely a line of future work. And the thing that's kind of shocking about this is that even without these sort of active strategies, this set of psychophysics is replicated pretty well. But we expect that this is going to be important in the long run to build the most accurate model we can.
Thanks, Andrew. Ramon Nogueira asks, similarly to convolutions being a key element of neural nets for vision, have you identified the most important structural elements of your architecture?
So we haven't
for
looked
this
in
particular
detail at
task?
that. The way we actually decided on these architectures was we performed a really large random architecture search and then picked the things that performed best on the natural environment task. And these were all convolutional neural nets. They're pretty small in comparison to like ResNet or something like that. And one thing we did find is that the kernels were almost never square. They tended to really elongate in time. So, I mean, we think that's important, but we haven't directly identified any specific things that are as important as convolution is for vision.
And for clarification, Andrew, these are conv nets applied to the spectrogram, or these null frequency spectrum.
So it's a nerve gram that comes out of the
representation,
Cosyne model. So it's not quite like a Mel frequency spectrogram or something, but it's along those lines. It still has that same sort of frequency decomposition that you'll see in a spectrogram.
But it's a two-dimensional spectrogram of the source.
And the two channels,
that is then fed
and then the
to
two channels are put into the channel dimension, basically. So the left and right,
And you're performing two-dimensional convolutions basically on those objects, yeah?
Mm-hmm. That's correct.
Okay, great. Thanks for clarifying. So Anna asks, does the network still perform the localization task well if it's trained on non-natural sounds? And I think you had a slide that addressed that, yeah?
Yeah. Let me go back to that real quick. So this is probably the highest level version of it, this one right here. And so this is the unnatural sounds task. And so it definitely performs worse. But what's interesting is kind of where it performs worse. It seems that it's mostly on this specific task. It does though. It does perform fairly well on the naturalistic task. Like it still learns its task pretty well, but it's not quite as good as the most normal model.
Okay, great. Thanks. So an anonymous person asks, how good are humans at sound localization in noisy environments, and how worse are your current models in their predictions?
Yeah, so that's actually an interesting question. It's the psychophysics literature isn't super clear on that. There aren't a lot of data sets that really test in complicated auditory scenes. It seems that humans are pretty good at this task, and our model definitely has some room to improve, especially, as I mentioned previously, in the front-back confusions. There definitely is a little bit of room to improve there. But there's actually a fair amount of behavioral work to be done there to really characterize that accurately. And it's just harder because you can't really do it in a sound booth. You need a much more complicated setup.
Great, thanks. Dory asks, can you read out the position of the microphone in the room from your network without explicitly teaching it?
Can you read out the position? Well, that's kind of what I had mentioned earlier. I think you mean, sorry, I'm not totally sure I understand the question. So without training the network, can you tell where the sound source is? Oh, I see what you're saying. So we haven't tested that. We do move the position of the listener around significantly in the virtual acoustic world, so it has to learn to be robust to that, because if you're near a wall or something like that, you get a totally different series of echoes and reverberation than you would if you were not in a corner, let's say. But
sure.
But we haven't explicitly seen if we can try to read out any of those environmental factors directly from the network.
Okay, great. Thanks a lot, Andrew. I think we're going to wrap it up here, and I'm going to welcome the next speaker. Thanks. This was super interesting.
Thank you.
All right, take care.
Yep, you too.
All right, we're going to welcome our final speaker of this session, Madineh. Hi, Madineh, how are you?
Hi, good. How are you?
Great. Yes, thank you for sharing your screen. So Madineh is our final speaker, and she's going to tell us about sinusoidal transformations of the visual field on the cortical surface. You can go ahead whenever you're ready, Madineh.
Great. Thanks, Demba, and thanks to the other organizers for putting together a really great virtual conference. I want to start by thanking my co-authors in case I ran out of time, Quo, Nicole, and Rachel, and especially David Fitzpatrick, the PI. So responses in the visual system are organized in a couple of different maps or large-scale organizations. The first is the retinatopic map, where the visual field incident on the retina is mapped onto the cortex using a simple point-to-point transform that, albeit deformed or magnified in certain areas, preserves local and global relationships, and this is known as a conformal mapping. The other type of large-scale organization is functional domains, where different pieces of cortex have different sensitivity to different visual features. And probably the most striking example of this can be found in primate V2. So that's this thin strip, the tip of which is visible here, that has these different segregated interleaved stripes that are sensitive to things like orientation, color, motion direction, disparity, etc. And we don't really understand why these functional domains are segregated and interleaved in this way, but one idea is that their interleaving ensures that each part of the visual field on the cortex here has access, equal access to these different features all along the visual field. I'm going to show you some data from another animal model, the tree shrew, that suggests a simpler explanation. We think that these functional domains in V2 reveal that central and peripheral parts of the visual field have specialized processing, and that the retinatopy map in V2, the key discovery that led to this conclusion is that the retinatopy map in V2 isn't simple like in V1. It's striped, resulting from a sinusoidal transform of the visual field. And so these striped functional domains are really reflecting a striped retinatopic map together with central and peripheral portions of the map having different feature sensitivities. So here's a little tree shrew. They look like squirrels and have smooth brains that are ideal for imaging and a pretty well-developed visual system. I put small cranial windows, about 5 millimeters, over the border of V1 and V2, this thin area next to it, and I use GCamp and normal bars to map azimuth or elevation axes of the visual field. And the V2 border over here maps the vertical meridian, the most central part of the visual field. And just to orient you, I'll point out that that color is red in our color map. So here's some data. This is the elevation map. These are single bar activation patterns as a bar moves from the lower part of the visual field to the upper part. And you can see basically a corollary bar-like activation pattern in V1 and V2. It's mirrored, it moves in the opposite direction, but basically there's a simple mapping of the stimulus on the monitor on the cortex in both V1 and V2, so no surprise here. For azimuth, things look pretty different. In V1, again, there's a corollary active bar, V1 on the right here, that moves with the bar until it's no longer visible in the window because it's too peripheral, the bar is too peripheral. But in V2, there's multiple active regions instead of a single one, and they seem to sort of diverge and converge as we move from central portions of the monitor to peripheral portions. And it's not clear immediately what sort of transform takes this input pattern of the visual field on the monitor and produces this striped pattern on the surface of V2 with multiple gradients between periphery in blue and center in red. But this doesn't have to be a tough mental exercise because we can actually measure this transform by drawing lines on the cortex. So this is a line on the elevation map along the long dimension of V2, and it produces, if we take the preferred elevation values under the line, we get another line with some sort of wiggles, but basically a linear transformation. The same line on the azimuth map produces the sinusoid with oscillations that carry you from central portions of the visual field to periphery and back. And putting these together, you can see that as you walk across V2, you get this sort of snaky sinusoidal pattern where you reverse several times between center and periphery, and these red points where the direction of the path changes correspond to the middle of these central and peripheral stripes where the direction of the color gradient changes. So the point is that these stripes on the azimuth map are really just a manifestation, they're a byproduct of this sinusoidal transform. And to see this a little more clearly, I'm going to show you this animation where I'll actually build the map in V2 from the color map in the visual field and the sinusoidal transform. So for each point in the visual field, as I go along this path here, I'm going to put a thin bar in V2 of the same color. And now it should be clear that as this path reverses, it's going to produce this series of stripes in the azimuth map in V2. And we'll do the same, but just change the color map of the visual field for elevation, but maintain the same transform. And now you'll see we'll get a single coarse map of elevation, albeit with some wiggles resulting from these reversals. And I want to point out here that this sort of sinusoidal one-dimensional coverage of a two-dimensional field is a known solution. So for instance, if you wanted to cover a two-dimensional field like your lawn with a lawnmower, a one-dimensional operator, under some scenarios, this path would be the optimal path to take. So what we found is that although V1 in the tree shrew, like many other animals, has simple retinotopic maps along orthogonal axes, in V2, the maps are along the same long axis. So the two axes of the visual field are no longer maintained orthogonal on the cortex. And this is a real departure from every other sort of retinotopic map we're used to seeing. And you might ask then why these two areas that are right next to each other might have such different transforms of the visual field. And I don't really have time to get into it here, but one obvious answer is that they have different shapes. Area V2 has a long aspect ratio, is thin. And we did some modeling. You can check out the preprint. There's a section in there that shows that you can use a self-organizing model with the same type of parameters and physical constraints to produce either simple linear retinotopy maps at low aspect ratios resembling V1. And if you change nothing else but stretch this area, you'll start to produce these striped azimuth maps. So as the shape of the area changes, as it becomes very elongated, the optimal visual field transform is no longer a linear conformal transform. That's the point here. So the second question is, well, how does this get physically implemented in the brain? V2 doesn't actually receive direct retinal input. It receives most of its visual field information through V1. So the most parsimonious explanation would be that the simple map in V1 gets transformed to the striped map in V2 from a sinusoidal wiring pattern between V1 and V2, which sounds crazy, but we can actually test this. So we create known input patterns in V1 using colored tracer injections, and then we compare the pattern of axon terminal fields in V2 to predictions of the sinusoidal transform. So here I've made a coarse azimuth map in V1 by injecting yellow tracers along the central representation near the vertical meridian border, and green tracers along the more peripheral portions of V1. And I'm going to now run the simulation as before. And you're going to see two sets of holes. So let me pause this. These thin holes between the yellow central representations are just because we don't have label where the path reverses. So I didn't inject label right up to the border between V1 and V2. The thicker holes, these guys, are again because I don't have label in the most peripheral portions of V1, which are actually sort of tucked in the midline. I can't access them. So that explains the two sets of holes that we see here. But otherwise, the point is that as this path reverses, the order of the colors and the axon patterns in B2 changes. So you go from green to yellow, and then there's a hole, and then yellow to green. And that's exactly what we see with the axon terminal. So this data suggests that it's the pattern of connections between B1 and B2 that actually implements this transform. Okay. And I've mentioned a couple times now that the sinusoidal transform has multiple path reversals, multiple gradient reversals within a single mapping of the visual field. And it's important to recognize this because linear retinotopy maps that we're used to seeing don't have any reversals. And in fact, we use this idea to put an area border every time we come against a reversal in visual field maps. So this is seen in practice here. This is anatomical data from the flattened mouse visual system where tracer injections are made in V1. And for instance here, where the axon terminal colors go from red to yellow and then yellow back to red, we would put an area boundary there. But if the underlying retinotopy isn't linear, maybe it's sinusoidal or some other transform that includes reversals, then even small injections in B1, like here in the tree shoe, will produce multiple patches of label with several reversals in between. So if I were to put an area border every time I came against a reversal in B2, then I would get all these little areas, each of which only maps a small portion of the visual field, and I would need all of the reversals together to actually produce a single map of the visual field. So my point here isn't that all of these higher visual areas in the mouse are a single V2 or that there's sinusoidal retinotopy just like in the tree shoe, but it's just that we need to be aware of other visual field transforms and not rely so completely on reversals. So we also verified striped retinotopy maps using the pattern of callosal projections, inputs from V1 of the opposite hemisphere that bring information from the other eye, from the other visual field. And callosal projections are known to terminate in regions of the cortex that map the vertical meridian, that central representation. So here I'm showing you in this map of the azimuth that the most dense callosal termination patterns overlap the central stripes in V2 and the region near the V1, V2 border where we know there's a vertical meridian representation. And if we zoom out and look at the whole flattened section, again, you see striped callosal projections in V2 reflecting the underlying striped retinotopy map and a single active region in V1. So the pattern of callosal projections can be a surrogate for the type of retinotopy map. And this is convenient because it's hard to measure functional retinotopy maps in animals, especially those with folded brains, but it's relatively easy to measure callosal projections and then post-mortem flatten the brain. And I mentioned this.
Just a heads up, Madineh. Yeah. Madineh, heads up that you have a couple of minutes, yeah.
Okay, thanks. I mentioned this because callosal projections in V2 aren't unique to the tree shrew. They've been observed in a couple different animals, most notably primates. So here's or macaque. Here's a flattened section of the macaque where you see these striped patterns of callosally projecting neurons in area V2. So this suggests that macaque V2 might also have a striped representation reflecting a sinusoidal transformation of the visual field. Oh, and the other thing this tells us, this striped pattern of callosal projections, is that these regions might be more sensitive to binocular stimuli or, say, horizontal retinal disparity stimuli. And that's exactly what we see when we measure binocularity sensitivity, horizontal disparity sensitivity, and sensitivity to grading orientation. So there's two things I want to point out quickly here. One is that there are functional domains. There are segregated sort of stripe-like functional domains for each of these visual features. And the second is that their pattern is correlated with the pattern of the retinotopy map. So when you put these together, you can see that the central parts of the map have different functional properties than the peripheral parts. So these two, the binocularity and disparity, make sense or consistent with the pattern of callosal projections. The orientation is a little harder to explain. We can discuss it in questions, but we think it has to do with the pattern of orientation that's elongated near the V1, V2 border. And this brings me back to the top, to the monkey. We think that the monkey V2 also has a striped retinotopic map, and that different regions of these stripes, so for instance, the foveal stripes have different feature sensitivities. For instance, they might be more sensitive to color, and that's why we get these striped feature maps. Okay, and I'll wrap up. So we have two main findings. The first is that there's a sinusoidal transform of the visual field in area V2 of the tree shrew. And this is important not because of the sinusoidal transform, but because it opens the door to looking for and being aware of other non-conformal transforms of the linear field and not of the visual field, and not assuming linearity everywhere, which can get you into trouble, for instance, with delineating areas. We also found that there's functional specializations for central versus peripheral visual field, and that this might explain the segregated feature maps in V2. And I don't have time to go into it, but this may also suggest that there's actually a retinotopic underpinning of the parallel dorsal and ventral streams in the brain, where foveal or more central parts of V2 give rise to the ventral stream, and that bias is carried to other areas like area IT. All right, and with that, thank you.
Madineh, apologies to interrupt you. Yeah, so we have some time for the Q&A session. Thanks for an awesome talk. Let me just get into the first question. Is there a good reason why the two axes are treated differently in V2? That is to say, why is it not the other way around?
Yeah, that's a great question, and it has to do with the vertical meridian at the boundary between V1 and V2. So because the vertical meridian is along this elongated boundary, that has a greater influence on the elevation map and constrains it, where the azimuth map is more free to sort of fold in on itself. So in the paper, we go through this. The model was actually nice in helping us figure that out, but we think it's the vertical meridian influence.
Awesome, thanks. There's a question here by Arrow who asks, do you have any thoughts regarding the functional implications? That is to say, are there stimuli, for example, that would be easier or harder to detect/discriminate given the sign map as opposed to a Cartesian map?
Yeah, that's a good question. I don't really know as far as, why is this beneficial? What are the functional implications of this? What would be the optimal stimuli? I think that obvious guess is any sort of stimuli or behavioral task that involves bringing central and peripheral portions of the visual field together, things like saccades or reaching and grasping behavior. But this work doesn't really help us get there. We are starting to characterize the receptive fields in central and peripheral stripes of V2, and I think that'll help us find some answers.
Okay, awesome. There's a question that asks, how many copies or passes through the azimuthal axis are there in the full V2?
Yeah, we see about 6 full stripe sets. So something like 11 to 12 reversals and 6 full stripe sets. In the V2 of the tree shrew, I think that number will be different depending on the exact area and shape of V2 and other animals relative to that of V1.
Thank you. There's a question here that asks whether the tree shrew has cytochrome oxidase stripes in V2, and if they do, how do you think these retinotopy findings map to the stripes?
I love that question. Unfortunately, the tree shrew doesn't label nicely for cytochrome oxidase in V2, so we don't see evidence of stripes. But I think that cytochrome oxidase labeling in the primate actually correlates with the visual field. And there's a little bit of data on this because callosal projection patterns, which are sort of a corollary for the vertical meridian, line up well with the pattern of with the pattern of cytochrome oxidase. So my hypothesis is cytochrome oxidase correlates with visual field.
Awesome. I think we have time for a quick answer to a last question. How would predicted connectivity differ between a sinusoidal map and multiple linear maps with reversal?
Yeah, so we have actually looked at this a little bit. If we inject within a central or peripheral compartment of V2, we see sort of harmonic patterns of connections. So if we inject in the central region, you have a hole where the next door peripheral stripe is, and then you have these sort of harmonic jumps of connectivity. So the connectivity pattern maintains connections between nearby regions of the visual field, but not nearby regions of the cortex.
Awesome. Thanks for an awesome talk, and thank you very much. This marks the end of our session. Let's thank all of our speakers. Thank you.